I'm Kathleen Martin. I'm the chair of Vascular Discovery 2024, and we are really excited to have kicked off the meeting this morning here in Chicago. And I'm really thrilled to be here with one of our plenary speakers in our Hot Topics in Cardiovascular Disease session, Dr. Ken Walsh, who's a professor at University of Virginia. He's also my PhD thesis advisor, little known fact. So it's really great to be together again after all these years and to see the amazing directions your work has taken. It's really inspiring, and I was excited to have you at this meeting to tell us all about clonal hematopoiesis and the relationship that it has to aging as a risk factor for cardiovascular disease. So if you could explain a little bit about what clonal hematopoiesis is and why it's important for aging. You gave such a great talk this morning, but this is such a big concept that we would like everyone to know about because it's really changing the way we think about cardiovascular disease risk.
Sure, Kathy. Thank you for having me here, and thank you for the invitation being here. It's really wonderful to see how you've developed and how much you've accomplished in the years since you left my laboratory. Not that long ago. So our recent interest has been on the area of clonal hematopoiesis, which basically is a consequence of the somatic mutations that we acquire as we age. I like to say that there are two types of mutations. There are the mutations that you're born with, and then there are the mutations that we develop with age in all of our tissues, and but individually in our different cell types. And it's been recent advances in technology and a couple of landmark papers that came out in 2014 that really indicated that these somatic mutations are contributing to features of biological aging, age-associated diseases, and of course cardiovascular disease. And when these papers came out in 2014, you know, we by Jaiswal et al. And Genovese et al. Back-to-back papers in New England Journal of Medicine, we read those papers very carefully in the laboratory, and we said, even though we were working on what we thought were some pretty interesting projects, we said, you know, this is a new mechanism of cardiovascular disease. And you got the sense from looking at those papers that this was going to maybe capture some of the importance of biological aging as the major risk factor of cardiovascular disease. And so we basically initiated a program on that. If you want me to go into a little bit more background on what is clonal hematopoiesis?
Sure.
Okay, so
it's really frequent as people age.
Yeah.
It's uncommon in young people, and as you explained, by the age of 70, almost everyone has some degree of clonal hematopoiesis.
So when we're conceived, we have the most homogeneous genome that we will ever have, and then you know, as we age, you know, during embryogenesis and as a child and going into advanced age, we just accumulate mutations in all cell types in our body. And what will happen is that some mutations in what we refer to as driver genes can give a selective advantage to the cell type. And so basically, as we age, because of these driver mutations that lead to these clonal events, we basically become a patchwork of evolving clones, essentially, as we age. And you know, these clones not only diminish the diversity of the cell types in our immune system, which is where we study this largely, but the corrupted immune cells, you know, we're able to now study in the laboratory and demonstrate that they are genetically corrupted and they don't function in a normal physiological homeostatic manner, and they alter immune cell function. Sometimes it's more pro-inflammatory. Sometimes it diminishes the ability of immune cells to conduct immune surveillance, and they contribute to cardiovascular disease and other diseases. So there's been a lot of activity literally around the world where people are measuring clonal hematopoiesis. As I pointed out in my talk, the methods of measuring clonal hematopoiesis are still evolving. We're still really bad at measuring clonal hematopoiesis. You know, if you really want to admit it, you know, there's still a lot of gray areas, a lot of really dark areas where we're trying to figure out, you know, what is the driver gene? What is a clinically relevant clone size? How do different driver genes and different clones, you know, contribute differentially to disease? Because developing clonal hematopoiesis is a very personal thing. my clonal hematopoiesis that I am developing is going to be very different from the clonal hematopoiesis that you have. And if I speculate, I just think that it's going to take us down on these different trajectories of disease, like why it is that some people are going to basically get cancer and other people are going to get some form of like or heart failure or atherosclerotic cardiovascular disease or peripheral vascular disease or something like that, like why, do we show so much diversity in disease? And I think that in part, the clonal hematopoiesis, because it's so individualized, is basically taking us down these different disease tracks as we grow older.
This is really exciting that the ability to measure these mutations in hematopoietic stem cells that cause these dramatic changes in the behavior of the immune cells affecting so many diseases, including cardiovascular disease. This is really an exciting diagnostic and a new era for personalized medicine. As you said, if we can really understand the nature of these individual mutations, we can start to develop personalized therapies.
Yeah. And actually, a good example of that was what we've learned from the CANTOS trial. And I pointed that out in the talk, and I'll mention it again because I think it's so important, is that CANTOS was looking at canakinumab, a neutralizing antibody to IL-1 beta, in a very large subset of its dose escalation, placebo-controlled. And they found that there was a signal that individuals at high-risk cardiovascular disease on canakinumab showed a reduction in major adverse cardiovascular events. Subsequently, they went back and reevaluated the 4,000 of the 10,000 individuals that were enrolled in CANTOS, and they assessed those individuals for their clonal hematopoiesis. And it turned out that individuals that had major clones in the driver gene that's referred to as TET2, something that actually
one of my
you
favorite
study.
genes.
Yes, you're working on Hot Topic. And it actually turned out that TET2 is one of the really, of all the different driver genes, it's very robust in terms of turning on IL-1 beta. And in the paper that we wrote all the way back in 2017, we actually speculated that individuals that had TET2 clonal hematopoiesis would maybe respond best to an inflammasome inhibitor or a neutralizing antibody to IL-1 beta. And in fact, that's what was found in that subsequent analysis of CANTOS. It turned out that individuals that had TET2 clonal hematopoiesis had something on the order of like a 60% response rate in terms of reduction in major adverse cardiovascular events if they had a mutation in TET2 compared to individuals that did not have that mutation. In fact, you can almost parse the data out and make the conclusion that the only people that really responded in that trial to the canakinumab were individuals that had TET2. And I know that there's a lot of activity right now to pursue that, both in academia as well as, probably pharmaceutical firms are looking at that as well.
That proof of concept of personalized medicine based on clonal hematopoiesis was really a huge breakthrough. And I'm sure it's only the beginning of future therapies that will be based on clonal hematopoiesis and individual driver mutations. That's really fundamental and exciting. And the other major discovery that you told us about this morning that's so fascinating is how somatic mutations, specifically loss of the Y chromosome, might be responsible for the difference in longevity between men and women that's seen not only in the US, but in populations around the world. Could you tell us a little bit about that work?
Sure. So that grew out of the clonal hematopoiesis work. And it actually is a version sort of clonal. It's genomic instability in the hematopoietic system. And one of the things that we were struck with when we struggle with, where we've got to go in the future with clonal hematopoiesis, we've got to get better at sequencing. We need more granularity in terms of the different driver genes. Before I get to that, one of the things I can point out is that, the early papers suggested that individuals really just get a single clone. But if you do ultra deep error corrected sequencing, which is what I recommend that everybody does, even though it's very expensive, so you can only do it on small, well-defined cohorts, you see that everybody's got a very complex landscape of clonal hematopoiesis. Everybody develops multiple clones that if you look at this, basically at a deeper level. But even with that progress and even with that understanding, we realize that there are still some individuals that basically have clonal events, but we can't identify what their driver gene is. And so you can look at the amplification of passenger genes in these individuals, and you can see that they are being basically amplified. And you go, but what's the driver? I don't see a TET2, I don't see a DNMT3A, I don't see a mutation in P53 or any of these things. And so we started thinking about it. And of course, like with everything, you go after the low-hanging fruit. And we were kind of aware, we started reading other papers on genome instability. And it was really from the landmark work that we built off of by Lars Forsberg at Uppsala University. He's sort of an evolutionary biologist, and he became interested in the issues of male sex chromosomes and made the first observation that men that have accelerated loss of the Y chromosome, that's associated with an increase in mortality. And as you mentioned, that's particularly important because loss of the Y chromosome is associated with this big difference between males and females in terms of overall longevity and, of course, certain types of diseases as well. In Western industrialized types of nations, the difference between a female lifespan and a male lifespan is 6 years. Females live on average 6 years longer than males. And most people think that a lot of that mortality is being driven by things that men did early in their life, maybe driven hormonally, high risk behaviors and those types of explanations. And of course, that is an explanation and that does have an effect. What's really surprising is if you look at the mortality curves, that most of the divergence between males and females happen in our post-reproductive years. And women, go through menopause, and men, their levels of that harmful testosterone go down as well. So women are losing the protective hormone, and men are losing the sort of the destructive hormone. Yet women outlive men by 6 years. And so much of that extended lifespan is happening in the last years. Women biologically probably age better than males. And so that's been associated with loss of the Y chromosome. Loss of the Y chromosome can maybe explain half of that mortality at an epidemiological level. And so we've basically started building animal models and also doing additional work in the cardiovascular space, looking at particularly diseases where there are strong sex biases. We've been looking at the sex chromosomes there to see if that can explain, some of this sex-driven mortality and this male-associated biological aging.
Your discovery of key genes on the Y chromosome and their homologs on the X chromosome is really interesting and likely an area of important investigation in the future to understand this difference in aging between men and women and perhaps target therapy toward these genes.
Right. Yeah. So if you ultimately want to take it to a reductionist approach,
right.
You want to just say, a whole chromosome, even though the Y chromosome is a very small chromosome and it's a gene desert, but our work showed that there are genes that are on the Y chromosome that are important for normal immune cell homeostasis. And we wanted to identify what those genes were. And, in terms of the readout that we do in our mouse model, it could be another gene for another disease or whatever, but it sort of came down to one of the genes, which actually encodes for an epigenetic regulator, which is referred to as UTY. And females have a copy of the UTX gene. And so, it and of course, it's, as I think I mentioned, it's an epigenetic regulator, but it gives us a handle, to study this at a more mechanistic level, getting into cell culture, doing all the single cell types of analyses and all the different types of highly molecular analyses that we're capable of doing today. Maybe we can get some handles in understanding this better and maybe even coming up with therapies.
You had mentioned that your study of clonal hematopoiesis has changed the way you approach science in general, kind of flipped it around starting with the humans.
Yes. As you will remember, when you were in my laboratory, everything was cell culture, and then we strove to get things into mice. And then, if you've been doing this a while, people will start to anticipate like, well, what's really the clinical relevance of your experiments, and you start striving to look for these relationships in human cohorts. And I'm in a fortunate position now, and being where I am at the University of Virginia and having the opportunity to work with cardiologists at the University of Virginia, literally around the world, we go the other way now. We start, with a clinical measurement. So for example, looking at loss of Y. And in fact, I'll just let this drop also. We also look at loss of X. Women will lose their inactivated X chromosome as they
Really?
age as
Wow.
well. Yeah, yeah. But there's like really no good epidemiology on that right now. But we've developed and validated the assays to look at that. And we go, okay, but first we want to see that it's associated with a disease. And then we'll build a mouse. And then if we get all this, if all these things are lining up, then we're going to study it, in a deeper mechanistic level. But it's really being driven by the human data now, rather than starting with the kind of the molecular biology initially. So it totally flipped it around.
That's really great and adds so much translational relevance and brings us so much closer to the clinic, to prevention, to therapies. Thank you again so much for coming to the meeting and for sharing your exciting work with us. This has really revolutionized how we think about aging as a risk factor for cardiovascular and other diseases. And I can't wait to see what you'll discover next.
Okay. Thank you, Dr. Martin. It's really been wonderful to be here. A great meeting. Congratulations.
Thank you.